You have used one of these, and you have certainly used one of these. In a science lab, we need to do the same thing to move liquids around, but we have to do it with more precision, and we use a pipette for that. Pipette refers to the tube itself. The word actually comes from the French, meaning little pipe, and we also use it as a verb, just to make things more complicated. The first pipettes, which are still in use today, are called Pasteur pipettes, named for this guy, Louis Pasteur, a French chemist and microbiologist, who spent his life studying diseases and developing vaccines and what we now call pasteurization, collectively saving millions of lives, and in the process, he needed a device to move liquids around without contamination, and thus the pipette was born. But basically, they're just tubes that we use to push and pull liquids up and down by creating pressure gradients. So they're just fancy straws, really. Now, in a lab, you would never ever use a straw, okay? I was just explaining to you the basics of how these things work. Now, a real pipette is going to look something like this. You always want to read what it says. It will tell you what the volume is that it will dispense and what the tick marks represent. It'll also tell you what temperature it was calibrated at, because remember, volume is temperature dependent. This is a 10 milliliter pipette, and it's telling you that between each numbered increment that represents 1 ml, the tick marks are a tenth of an ml. This bad boy is 100 milliliters. Each numbered increment is 5 mLs, and then each small line represents 1. So when you use these things, you want to attach a controller, it's going to be a pump or a bulb, onto the end, and then place the pipette vertically in the solution. And then don't let that tip touch the bottom, by the way. Slowly go ahead and use the bulb or the pump to draw up the solution to aspirate. Keep your eye at the same level as the meniscus. That's where you want to read it. To dispense, you're going to do the reverse with the bulb, or in this case, I'm depressing a plunger. Now, if you need to move small volumes around, you're going to reach for a micropipetter. We usually use the term pipetteman, which is actually a trademark, kind of like Xerox. You know what I mean. Step one is to select the correct model of pipetteman to use, and that's going to be determined by the volume that you want to dispense. So the top of the push button or the plunger has what we call a P number. That's the maximum volume in microliters that can be dispensed by that model. Now, we're talking about microliters here, so quick review, 10 to the minus sixth. 1 microliter equals 0.001 milliliters. 1,000 microliters is 1 ml. Okay, so when we look at different models of pipetteman, and there are others, by the way, the P number just refers to the maximum volume in microliters that can be dispensed by each model. So hopefully that's not too tricky. For each model, there is an adjustable range. So this means that you can use the plunger and the wheel to adjust what volume you actually want, but there is a recommended range for each model, which is narrower, meaning that's the range that's going to give you the most accurate result. This is also helpful. It's the smallest increment that each model can dispense. Okay, so you're going to select a model based on the volume that you want, and then you're going to adjust the volume by dialing it in, and you read the window to see what volume you're getting. So with the plunger, you can adjust big changes in volume. The thumb wheel is what you'll use most of the time for fine adjustments, and you're going to be reading in that window. Now, the place values vary depending on the model you're using. Remember the place values? Do you remember those? I just flipped it, so don't panic. Okay, so I'm going to compare a P20, P200, and a P1000, dialing in 2 different numbers for each one, and we're going to figure out what volume you actually get. Now, by the way, I have used red where the actual models use red, so I didn't do that randomly. Okay, that's what it's going to look like for you. When you dial in 015 on a P20, what does that mean? The line represents a decimal. Okay, so you've got the tens, the ones, the decimal, and then the tenths place. So when you dial that in, what you actually get is 1.5 microliters. What about 100? So try that one. What would that give you? So that's 10 microliters. Okay, what about the P200? When you dial in 015, what do you get? The P200 does not have a decimal, so you're looking at hundreds, tens, and ones. So 015 gives you 15 microliters. So you try 100. What are you going to get? Try it. Yay, 100 microliters. What about the P1000? Now, with the P1000, they do use red for the thousands place. So 015 on a P1000 gives you 150 microliters. And 100 on a P1000 gives you, try it, 1,000 microliters. This will take practice, okay? In the beginning, your head will hurt, but that's normal. Just keep going. As you keep using them, it will eventually click and it will make sense. Step 3 is to mount a tip. So go with the pipetteman straight into the box of tips gently and rotate a little bit. You do not need to slam the pipetteman into the box of tips. Next, you're going to have to understand that the plunger has 3 different stops. At its highest point, we call it a rest position. Then we have a first stop, and then we have a second stop or a purge. So when you're using it, in order to prepare to draw the sample, you're going to push the plunger to the first stop. Then you're going to insert the tip into your sample and aspirate. That means you're going to suck up the sample by allowing the plunger to go to the rest position. Then you're going to move the tip into whatever tube you're going to dispense into. And then on an angle, you're going to dispense, meaning the tip is at an angle, and you're going to push the plunger to the first stop. Then you purge. That means you push more. You're going to feel that resistance. And what you're doing is dislodging any little droplets that might be stuck onto the side of the tip, and you're going to get those out into your sample. And finally, you're going to eject the tip by pushing this other button at the top, and make sure you eject into an appropriate container. So let's do it. So here I go. This is my pipetteman. I just added a tip. And now I'm going to depress and then insert the tip, slowly aspirate, and then go onto the side of the tube I'm dispensing into. Here's the first stop and then the purge. That's hard to see, so let's look at that again. First stop and then the purge. All right, how about a point of view? So here I am. I'm very, very slowly aspirating, so I avoid air bubbles in there. And then to dispense, I'm going to go on the side of the tube, go all the way to the first stop, and then purge. And yeah, I want to thank Insta360 for the Go 3 for that shot because I couldn't resist. And don't forget when you're done to use this button to eject the tip into an appropriate container. Don't do this. It was a long day. I hope that was helpful as always.